Cascade impaction measures particle size in inhaled therapies. Air is drawn through stages, accelerating particles for separation. The next generate in low drug knots.
To test a metered dose inhaler, an analyst must actuate the device directly into a sampling apparatus. The resulting aerosol cloud is highly chaotic. It is a dense, fast-moving mixture containing both the active pharmaceutical ingredient and the inactive carrier formulation. Standard analytical tools, like optical or laser diffraction, measure this entire cloud indiscriminately. They capture the size of everything passing through the beam. Knowing the total particle size of the formulation does not prove clinical efficacy. A patient's outcome depends entirely on where the active drug particles specifically deposit in the respiratory tract. Laser diffraction cannot distinguish a drug molecule from a carrier particle. It simply reports the size of the total mass. To see the drug alone, we must physically capture its individual size fractions during flight. This requirement leads to cascade impaction. It provides the size-fractionated physical samples required to generate an aerodynamic particle size distribution exclusively for the active ingredient. The Cascade Impactor operates as a physical sorting mechanism, using controlled volumetric flow and particle inertia to categorize the dose. At its core, an impactor separates particles based strictly on their inertia. In fluid dynamics, a particle's inertia is a function of both its mass and its velocity. The sorting process begins by drawing sample-laden air through the entire instrument at a fixed, constant volumetric flow rate. This diagram illustrates the internal mechanism. Airflow is forced through a narrow nozzle, accelerating directly toward a perpendicular collection plate. Because the plate blocks the path, the stream makes an abrupt 90-degree turn. This creates a threshold for separation. Particles with high aerodynamic mass possess too much forward inertia for that sharp turn, breaking from the stream to impact the plate. Remaining lighter particles successfully ride the airstream to the next stage. By repeatedly forcing the aerosol through these tight angles, the instrument uses a particle's own inertia against it to create a highly precise aerodynamic sieve. The industry standard architecture built upon this inertial theory is the next generation impactor. To isolate progressively smaller particles, the instrument faces an engineering problem. It must progressively increase the velocity of those particles at each stage, even though the total volumetric flow rate through the system remains fixed. This data visualization details the solution. Across the NGI stages, the total cross-sectional area of the nozzles systematically decreases. This restriction forces the air to accelerate. At stage 1, the airflow passes through a single, large 14.3 milliliter nozzle, moving just fast enough to capture the heaviest particles in the dose. By the time the air reaches stage 7, that same fixed volume is forced to accelerate through 630 individual micronozzles, capturing particles just a fraction of a micron wide. The sequence ends at the micro-orifice collector, which acts as a final physical filter, utilizing over 4,000 holes measuring a mere 70 microns across. The result of this engineered matrix is a series of size-fractionated samples, physically distributed across removable collection cups. The impactor hardware does not output aerodynamic data. The physical dimensions of the nozzles only define the cutoff diameters for each stage. The actual aerodynamic particle size distribution curve is generated off-board. Analysts recover the distinct fractions from the cups and subject them to targeted chemical assays, typically high-performance liquid chromatography. The precision machining inside the NGI serves a single function, transforming abstract fluid dynamics into quantifiable, chemically verifiable physical bends. A Cascade impactor functions as a near-perfect aerodynamic sorter. However, human airways are highly imperfect impactors. The throat and nasal passages dynamically filter and trap aerosol droplets long before they can reach the deep lung. Traditional Cascade impactors interface with inhalers using a simple, right-angled stainless steel tube known as an induction port. While this geometry provides a standardized test, it fails to replicate human regional deposition. This discrepancy creates a gap in vitro to in vivo correlations. To close that gap, researchers require anatomically representative inlets to gate the airflow before it reaches the precision sorting of the impactor stages. This anatomical pre-filtration is especially critical for evaluating nasal spray products, where regional deposition dictates whether a drug will act locally in the nasal cavity or be absorbed systemically. This compares the simple interior of a standard 90-degree induction port against the irregular, multi-chamber geometry of the Alberta idealized nasal inlet. It forces aerosol through distinct physical regions, mimicking the vestibule, the turbinate, the olfactory region, and the nasopharynx. By simulating the initial deposition in these specific cavities, this prefiltration ensures the impactor stages below only size the true respirable dose, the precise fraction that would realistically survive the nasal passage. Pairing precision inertial sorting with anatomical modeling produces a dataset that accounts for both the formulation's chemistry and the physical constraints of human respiratory physiology.